This nanopore-based detection method has demonstrated the potential to physically characterize and identify individual molecules. Working with metallo-nanoparticles, which is the purpose of this video, is challenging our understanding of what limits the measurement's accuracy and precision.
The main advantage of this technique is that this aperture has greater bandwidth than traditional lipid bilayer membrane setup, which lets us see faster results and more reactions in nanopore.
This technique was made possible because in the early 1990s, it was shown that gating in protein nanopores, that is, switching between different states, could be controlled, and molecules combined to the pore wall. This technique is being developed for commercial DNA sequencing applications because it uses minimal samples. It doesn't require fluorescent labels, and uses much longer read lengths than conventional methods do. This technique has also been developed to discriminate between polymers based on their size, with greater accuracy and speed than gel electrophoresis and chromatography.
First, prepare 500 milliliters of a solution of 1 molar sodium chloride and 10 millimolar monosodium phosphate in ultra-pure water as the electrolyte.
It is best to use fresh solutions in samples with this particular setup. In addition, we have found that high-quality, deionized water is very important for successfully forming membranes.
Next, combine 1 milligram of lyophilized wild-type monomeric S. aureus alpha-hemolysin powder with 1 milliliter of ultra-pure water.
Don't forget that working with Staph aureus alpha-hemolysin toxin protein can be hazardous. Follow the precautions in the MSDS when performing this procedure.
Next, prepare 4 milliliters of 5 milligrams per milliliter di-5-PC in N-decane in a glass scintillation vial. Cap the vial with